Okay, we'll start now. Hello, everyone, and welcome to the Medican Express webinar series. Thank you for joining us today. Today's topic is on novel predictive biomarkers that could overcome the resistance to endocrine therapies in breast cancers. As many of you know, breast cancer is both common and complex. The early stage disease is curable in many cases, yet advanced metastatic breast cancer remains largely incurable with current therapies. Different breast cancer receptor subtypes have very different clinical implications. For example, the estrogen receptor alphas, the PR and the HER2 can guide very different treatment paths for patients. Furthermore, the post-translational modification, including arginine methylations, add another layers that determine tumor initiation, progressions, and relapse. Today's sessions sits right at that intersections. So it's my pleasure now to introduce our speaker, Dr. Muriel Romances, the leaders of the endocrine resistance, methylations, and breast cancer teams at the Cancer Research Centers of Lyon. Dr. Romances is an internationally recognized expert on steroid receptors biology and breast cancer research. Her lab focuses on how post-translational modification, particularly methylations, regulate protein functions in breast cancers. Her works reviewed the critical roles of protein methylations in estrogen receptor signaling, uncovering novel mechanisms of non-genomic estrogen actions that impact tumor progressions and therapy resistance. And currently, Dr. Romances and her teams are pioneering the discovering of new molecular players in breast cancer tumorigenesis, with focus on translating these findings into predictive biomarkers for personalized cancer treatments. So for today's sections, just a bit overview, and Dr. Romances will speak for about 40 to 50 minutes. Then we will open it up for live Q&A. So please drop your questions into the Q&A panels in the Zoom chat at any times. We will collect them and address as many as we can at the end. And just to remind you that this webinar is being recorded. And with that, thank you again for joining us. Please join me in welcoming Dr. Romances. Thank you.
Thank you very much for the introduction and for the invitation to share with you today our results about the topic you announced, the role of the protein arginine methyl transferase in the steroid receptor signaling. And you will see that we started from basic science to clinical perspectives. And I just want to emphasize that I co-lead the team with a clinician, that is Dr. Trédan. And the goal of our team is really to bring our basic science to the clinic. So you may know that breast cancer is a very heterogeneous disease that is mainly classified in four subtypes based on the expression of different markers. Two of them are the steroid receptor, estrogen receptor, and progesterone receptor, and also the oncoprotein HER2. So these three proteins allow the classification of luminal tumors that express mainly estrogen receptor plus or minus the progesterone receptor, the H2 enriched also tumors, and you have also the TNBC, the triple negative breast cancers that do not express none of these three markers. This classification allows mainly to orientate different therapies. For the luminal tumors, it's mostly treated by hormonotherapy. For the HER2 enriched tumor, the therapies are tyrosine kinase inhibitors, but also antibodies or antibody drug conjugates targeting HER2. And for the triple negative breast cancer, very few targeted therapy are available for the moment, and they are mainly treated with chemotherapy. So let's just start speaking about endocrine therapy more in detail for estrogen receptor positive breast cancer. So these therapies are different upon the menopausal status and also the stage of the disease. For example, for the post-menopausal woman, they are mainly treated with aromatase inhibitor that will impede the conversion of androgen to estrogen, and so we will have no activation of the estrogen signaling pathway. For the premenopausal woman, they are mainly treated with anti-estrogen like tamoxifen that is classified as a SERM for selective estrogen receptor modulator, and its mechanism of action is the following. When the tamoxifen binds to the receptor, it will change the conformation, so the receptor will still be able to bind to the estrogen response element, but instead of recruiting co-activators, the change of conformation will allow the recruitment of co-repressors that will impede the transcription of estrogen receptor target genes. In the metastatic setting, one of the molecules that is used is another class of anti-estrogen, the fulvestrant, is of the family of SERD for selective estrogen receptor degraders. And this name because the mechanism of action is different from tamoxifen. The binding of the SERD to the estrogen receptor will trigger a proteasomal degradation. So fulvestrant has had a limited use in clinic because it's an intravenous injection. So this is the reason why now a lot of new oral SERD that are given orally have been developed by companies, and I have highlighted here two of them, elacestrant and camizestrant, that are still under clinical investigation, but a lot of other SERDs are really investigated and are in clinical investigation for the moment. So also we have all these therapeutics. We still have a strong proportion of the patient that will relapse by intrinsic acquired resistance. And the problem for the moment for the clinician is that apart from the expression of the estrogen receptor, we don't have any predictive biomarker of the efficacy of the different treatment. So we saw that to understand more about endocrine therapy, it's good to know more about the estrogen receptor signaling. It's a quite complex signaling because you have several pathways that are involved in the regulation of the receptor. On the left side here, you have three pathways that are called the genomic pathway because it involves the receptor that is a transcription factor and is located upon estrogen treatment in the nucleus, where you can have a direct binding of the receptor on its estrogen response element. You can also have, the receptor can be a co-regulator of other transcription factor, and the third pathway is activated by growth factor and chemokine, and through the activation of kinases like MAP kinase, by phosphorylation, the receptor is also able to be recruited on its estrogen response elements. But you have a fourth pathway, it's based with a small amount of the estrogen receptor that is localized close to the plasma membrane, and when it binds to SRC and PI3 kinase upon estrogen treatment, this creates a complex that will activate a pathway like ERK and AKT pathway that will also regulate cell proliferation and survival. And this pathway is called non-genomic pathway because it's involved this fraction of the receptor that is localized in the cytoplasm. You have to know also that another layer of complexity of estrogen receptor are this post-translational modification that was introduced at the beginning. This protein is highly modified, as you can see. Here you have all the different phosphorylation that are performed on the receptor, and most of this phosphorylation will impact the estrogen genomic signaling and the stability of the protein. You also have the hinge region that is located between the DNA binding domain and the ligand binding domain. It's highly modified by other modification you can see, such as sumorylation, acetylation, ubiquitination, and this modification regulate the stability of the receptor. And you also have three modifications that are shaded in gray here, that are modifications involved in the non-genomic signaling. For example, the palmitoylation here of the cysteine is important for the plasma membrane anchorage of the receptor. And our team identified that PRMT1, the arginine methyl transferase 1, is able to methylate the arginine 260 that is located at the junction between the DBD and the H domain. And I will go now through this modification. So PRMT1 is a member of the family of the arginine methyl transferases. There are 9 members, PRMT1 to PRMT9, and they are classified upon the type of methylation they perform. So first, all the PRMTs are able to monomethylate, adding a methyl group to the arginine residue of the target. Only the type 3 is able to monomethylate, only monomethylation, and after you have 2 other family that are classified upon the type of demethylation. So here you have asymmetrical demethylation because you have 2 methyl group on the same nitrogen, and you can have symmetrical dimethylation. PRMT1 is the main enzyme of the type 1, and PRMT5 is the main enzyme of the type 2. And this enzyme have been targeted by several inhibitors, like the GSK795 for PRMT1 and the GSK595 that are inhibitors that you can buy to make Metchem Express. So we use a lot of these inhibitors, but for example, for PRMT5, you have plenty of other inhibitors that are specific, and the private companies are developing a lot of these inhibitors, and some of them are currently tested in clinical trials. So now I will speak about first about the role of PRMT1, what we investigated in the non-genomic pathway. So several years ago, arginine methyl transferases were not very known by the researcher, and we decided to investigate whether ER-alpha could be a substrate for PRMT1 for different reasons. I will not go into the details today, and particularly because one of the consensus sites of PRMT1 is the RGG motif, and we saw that in ER-alpha we had 2 RGG motifs, one located at the junction between DBD and hinge region, and the other one at the end of at the junction between the E domain and the F domain of the receptor. And by in vitro experiment at that time, we first identified that the arginine 260 is the only methylated residue in ER-alpha. And thanks to this observation, we produced an antibody specific to this methylation site. And with this antibody, we were able to study ER-alpha methylation in cells. So we use the MCF7 cells that are express a lot of ER-alpha receptor, and they are sensitive to tamoxifen. And we were very surprised at that time to see that when we treat the cells with estrogen, methylation occurred very rapidly. You see here after 5 minutes of estrogen treatment, and was transient because after 15 minutes you see the decrease of the signal. Also, we are surprised to see that when we perform subcellular fractionation, even though ER-alpha is, as you know, it's mainly a nuclear protein, so it's mainly expressed in the nucleus, we saw that the methylation occurred only in the cytoplasmic fraction and not at all in the nuclear fraction. So to make a long story short, what we demonstrated in this work is that very, very rapidly after estrogen treatment, the first event was the methylation of the receptor, and after this methylation is a prerequisite for the formation of the non-genomic complex containing ER-alpha, SRC, PI3 kinase, and we also identified the focal adhesion kinase SRC as an important member of the complex, and this active complex is able to activate the downstream signaling, activating AKT involved in proliferation and survival. And as I showed before, I told you this complex is transient, and very, very rapidly we observe a demethylation that is concomitant with the disassembly of the complex. And several years ago, we identified, I won't go into the detail today, we identified that the arginine demethylase GMGD6 is the enzyme responsible for the demethylation and the disassembly and the decrease in the activation of the downstream signal. After having characterized this non-genomic pathway in cells, I have to tell that at that time, some people were saw that this non-genomic pathway did not occur in the real life in tumors. So we decided to investigate the expression of and the activation of this pathway in breast tumors. And for this, we were one of the first researchers using the proximity ligation assay technique at that time. We set up the technique in cells and after in tissue. And here you have the example of what we found. We were able to identify, to measure the interaction of E-alpha with PI3 kinase, E-alpha with SRC, and we also measure the methylation of E-alpha using an antibody raised against E-alpha and our methyl antibody to measure E-alpha methylation. And we decided to investigate also the phosphorylation of IKT by immunohistochemistry. So here you see two examples of the signal we obtain. One tumor that does not express at all the different members of the complex, and you see no activation of phospho-AKT. And here you have an example of a tumor that expresses a lot of E-alpha PI3 kinase, E-alpha SRC, methylated E-alpha, and also a very strong activation of AKT. And when we did this experiment on 175 tumors, we could make statistical analysis and we found a very strong correlation between each couple, but also each couple with the phosphorylation activation, the phosphorylation of AKT. So this result clearly demonstrated that this pathway is activated in the real life in tumors, in some tumors. So next, as you know, what is very potent with this technique is that you can count and quantify the number of dots per cells. We're able to perform statistical analysis to correlate the number of complex with the patient survival. And here the Kaplan-Meier curve show you for E-alpha SRC and E-alpha PI3 kinase, we found that when we have a high expression of E-alpha SRC or E-alpha PI3 kinase in red, it's really correlated with a worse survival for the patients, highlighting that this complex is really involved in the aggressivity of the tumors. More recently, in 2021, we confirmed these results in another cohort of 440 patients. Next, we thanks to our collaboration with Dr. Marangoni from Curie Institute in Paris that possesses a very huge number of models of patient-derived xenograft for breast cancer tumors. We had the chance to investigate the expression of E-alpha SRC and E-alpha PI3 kinase in models of acquired resistance to tamoxifen to mimic what happens in patients. And what we found in these tumors is that in the resistant tumors, we had an increase of E-alpha SRC and E-alpha PI3 kinase compared to the parental tumor. We next investigated the expression of the couple in cells, in MCF cells with acquired resistance to tamoxifen, and we observed the same result. In the TAM-resistant cell, we saw an increase of E-alpha SRC and E-alpha PI3 kinase. And interestingly, when we disrupt the interaction between E-alpha and SRC with a phosphopeptide, if you compare here the red curve, you have the resistant tumor treated with tamoxifen. You see that when we combine tamoxifen and this peptide that inhibit the interaction between SRC and E-alpha, we are able to partially restore tamoxifen sensitivity, showing that really confirming our result that this pathway could be involved in endocrine resistance. So based on all these results, we set up recently the Nestor project that aims at targeting E-alpha SRC with small molecules to circumvent resistance to endocrine therapy. So we have two approaches, an in silico screening to identify molecules able to disrupt this interaction, and a screen also of a library of chemical products specific to inhibit protein-protein interaction. And for the moment, we have identified 100 molecules based on the in silico screen, and we are still developing the automation of our screen, and the screen should start within a month. And we also have developed in vitro tests to really validate our hits in vitro, but also in cellulo. And I will show you for the moment one molecule that we have identified from the in silico screen. This molecule is able to disrupt E-alpha SRC interaction by B-molecular fluorescence complementation, but also by PLA. When we look at the interaction between E-alpha and SRC, we saw that this molecule is able to decrease the interaction. So this project is still ongoing. I can't tell more because we are waiting really performing our screen to identify it that we could validate further in preclinical model and maybe in clinics later. So now, first we started to work on PRMT1, and more recently we decided to also work on PRMT5, and you will see it's a very interesting molecule because PRMT5, I will show you today, has really a dual role in breast cancer, opposite effect according to the type of breast cancer you are working on. So PRMT5 is in the physiology, PRMT5 is really involved in all the molecular function. Here it's only a list, but it's not an exhaustive list. It has been demonstrated to be involved in signaling pathway, in the protein localization, in translation, splicing, transcription, DNA damage, genome integrity. So, based on this molecular function, it will also regulate a bunch of biological functions like the cell cycle, apoptosis, proliferation, differentiation, and so on. And this protein has an enzymatic activity, but works also in multimeric complexes. And one of the main partners of PRMT1 is MEP50. It's really its co-activator that will increase its enzymatic activity. It's a very important protein. You will see, we will speak about it later. When you look at the localization of PRMT5 in tissue, PRMT5 is mainly expressed in the cytoplasm. You have here an example with an immunohistochemistry. And this localization may be explained because PRMT5 does not possess any nuclear localization signal, but it possesses three nucleus export signals that can explain why it must go in the nucleus, but most of the time it goes out of the nucleus and it's localized in the cytoplasm. Interestingly, several publications show that the prognosis value of PRMT5 on cancer depends in its subcellular localization. In fact, you will see that cytoplasmic PRMT5 is mostly associated with aggressive phenotype. For example, here, if you look at the prostate, in the benign prostate or neoplasia, PRMT5 is mostly in the nucleus, but in prostate cancer, it's mostly localized in the cytoplasm. It's the same for the lung carcinoma and also for melanoma. For example, you see here, it's mostly nuclear in the normal epidermis and mostly cytoplasmic in the metastatic melanoma. So what happens in luminal breast cancer for the moment? So interestingly, if you look at the database at the mRNA level, for breast cancer, you find here in red that a high level of PRMT5 expression is really associated with a bad prognosis. But interestingly, when we perform an analysis in the team, investigating its expression at the protein level in 450 invasive breast tumors from patients treated at the Centre Léon Bérard, we found that PRMT5, as you can see here, can be exclusively cytoplasmic, but can also be expressed in the nucleus. And we are very surprised when we perform the Kaplan-Meier analysis here to find that if you look here in green, you see that a high expression of PRMT5 is really associated with an increased patient survival. So we confirmed this result more recently in 2 cohorts, and we found, so here it's in blue, so we confirmed that a high expression of nuclear PRMT5 is really associated with a better survival, but interestingly, we found that this result is only observable in patients treated with tamoxifen, but not with patients treated with aromatase inhibitors. So based on these striking results, we hypothesize that maybe there is a link between PRMT5 and E-alpha, as tamoxifen is a binder of E-alpha. So we perform mechanistic analysis and first try to investigate whether PRMT5 could bind to E-alpha. So we first showed in vitro by GST pull-down experiment that a GST PRMT5 is able to interact with E-alpha. We confirmed this result in MCF7 cells, by proximity ligation assay with an antibody targeting ER and antibody targeting PRMT5. And we found that with no treatment, we have a level of methylation in the nucleus of the cells. And in the presence of estrogen, we have a decrease of this interaction, and tamoxifen has no effect on the interaction. So next, we investigated whether E-alpha could be methylated by PRMT5. So we perform in vitro methylation assay with the different fragment of the receptor, and we found that PRMT5 is able to methylate E-alpha in the DNA binding domain that is represented here. So it's a highly structured domain with 6 arginine residues that are potentially methylated by PRMT5. I have to say that we spend a lot of energy, time, and money to try to investigate and to identify which arginine is methylated. But unfortunately, although for PRMT1, as you have seen before, we could identify the arginine 260, here, I guess because it's a very structured domain, each time we are mutating an arginine, we are losing some of the methylation. So we couldn't conclude, unfortunately, which arginine is methylated. But to circumvent this failure, we decided to use the proximity ligation assay using an antibody raised against E-alpha and a pan-methyl antibody recognizing symmetrical demethylation. So thanks to this PLA, we are able to measure E-alpha methylation by PRMT5. And you see here in this graph, donc, without any treatment, we see a level of methylation. Estrogen decrease this methylation, whether tamoxifen increases the methylation level of E-alpha. And here you have a control with the GSK, the G595 I presented before. In the presence of this inhibitor, we clearly strongly decrease the methylation, highlighting that PRMT5 is involved. We also perform siRNA. I don't present you all the details, but we are very, very sure about the specificity of the signal of methylation. So next, of course, we wanted to assess what was doing this methylation. So as I presented you before, I told you that tamoxifen induces the recruitment, the binding of co-repressors, transcriptional co-repressor to E-alpha. And one of the most famous is the protein SMART. So we decided to investigate the interaction between E-alpha and SMART in the presence of tamoxifen alone or combined with the inhibitor of PRMT5. So what we found is that we confirm what was already known, that tamoxifen increased the binding of E-alpha with SMART. But when we combine with the PRMT5 inhibitor, we completely lost this increase of interaction. We confirmed this result by co-immunoprecipitation. So here we immunoprecipitated SMART. So you see again, we have a very nice interaction with E-alpha in the presence of tamoxifen. And this was lost when we combined the tamoxifen plus the inhibitor of PRMT5. So the next question was to assess what was going on at the level of the chromatin. So we performed cheap experiments on 3 E-alpha target genes. So we treated the cells with tamoxifen, and we found, as was expected, an increase of the binding of E-alpha on its target gene. But when we combine tamoxifen here in green with the PRMT5 inhibitor, we didn't see any difference. So in fact, the binding of E-alpha on its target gene, it's not impacted by PRMT5 inhibition. However, when we chip SMART, we found that we still see the increase of SMART on the E-alpha target gene upon tamoxifen treatment, but we lost this recruitment when we combine the treatment of tamoxifen plus the inhibitor of PRMT5. So next, we just look at SMART. So we wanted to use a large-scale analysis to study E-alpha interactors. And we use again here the bimolecular fluorescence complementation. And we used cells expressing E-alpha and a library of 2,000 nuclear proteins involved in transcription. And we treated these cells plus or minus tamoxifen in presence or in absence of the PRMT5 inhibitor. And we found that 50% of the protein that are recruited to E-alpha upon TAT treatment are dependent upon PRMT5 catalytic activity. And when we used string analysis, we found that all the colored protein you see here are involved in the negative regulation of transcription, and we found a node around HDAC1, the histone deacetylase. So to validate our screen, we selected, we did the same experiment that we did with SMART, with HDAC1 and also with two other proteins, SIRT2 and SAP18. And we found exactly the same result that what we obtained with SMART. We have an increase of the interaction of E-alpha with these co-repressors upon TAP treatment, but we lost this increase when we combined the treatment of TAM with the PRMT5 inhibitor. So next, we wanted to study what happened in vivo if we could reproduce the result we found in MCF7 cells. So for this purpose, we engraft MCF7 cells in mice. We treated these cells with tamoxifen, and we looked at first at the methylation of E-alpha by proximity ligation assay using, again, the E-alpha and the antibody recognizing symmetrical demethylation. And what you can see is that we have a very low level of methylation in the nucleus of the control cells. But when we treat with tamoxifen, we have an increase, a significant increase of methylation that is quantified here. And we were very surprised also when we investigated the expression of PRMT5 and its coactivator MEP50. You see in the control tumor, you see that we have almost no expression of these two proteins in the nucleus. But when we treat the tumors with tamoxifen, we have a very strong increase of PRMT5 expressed in the nucleus. And interestingly, when we did the same experiment in tumors resistant to tamoxifen, we didn't see any increase of E-alpha methylation, and PRMT5 and MEM50 remain located in the cytoplasm. We didn't observe any translocation. We next decided to assess the level of E-alpha methylation and MEP50 expression in the tumors from the beginning. I presented you, we studied the expression of PRMT5. And you have example of what we found. We found a nice correlation between the expression of PRMT5, MEP50, and also the methylation of E-alpha. So here you have an example of a tumor that does not express PRMT5 and MEP50 in the nucleus, and you don't see any methylation of the receptor. And here you have a tumor that expresses, you see in the nucleus, a strong labeling of both proteins, and it's correlated with a lot of methylation, validating that this pathway on this methylation mediated by PRMT5 is occurring in breast tumors. So thanks to this result, we're able to propose this model. At diagnosis, the localization of PRMT5 expression is very important because we found that when it's localized in the cytoplasm, you have a low treatment efficacy with tamoxifen. However, when you have a high expression of the nucleus, we found a higher treatment efficacy. And in preclinical models, we found that even if you don't have a strong expression of PRMT5 in the nucleus, in some tumors, tamoxifen is able to trigger the shuttling of PRMT5 and MEP50 in the nucleus. When PRMT5 is in the nucleus, it's able to methylate the receptor, and this event is very important. It's a prerequisite for the recruitment of the big co-repressor complex that will impede the transcription of E-alpha target gene. At the contrary, estrogen, as I showed you before, disrupt the complex and decrease this methylation. More recently, I presented to you at the beginning that we have new molecule, new anti-estrogen, and we wanted to investigate whether this translocation was specific to tamoxifen or whether it can be generalized to other anti-estrogen. So thanks to our collaboration with Dr. Marangoni, we treated also PDX model with fulvestrant or camizestrant, and we found that in the sensitive tumor, we have also a very nice PRMT5 translocation in the nucleus for fulvestrant and camizestrant. Also in the resistant tumor for this drug, we don't see any translocation. So this result are very interesting because it seemed that this mechanism of the PRMT5 shuttling could be maybe generalized to other anti-estrogen, not only restricted to tamoxifen, even if the mechanism of action of this anti-estrogen are different. So now I told you it's a dual role. So we first saw that PRMT5 has maybe a good role in the luminal tumors. Now let's see what's happened with PRMT5 in TNBC, in triple negative breast cancer. So we found in the team that PRMT5 is a master regulator of the glucocorticoid receptor in TNBC. Just a small introduction for glucocorticoid and breast cancer. So you remember I showed you this scheme before with the triple negative breast cancer. And I told you that even if we have some novel targeted approach like PARP inhibitors and also immunotherapy, most of the tumors are still treated with chemotherapy, most of the patients, like doxorubicin, paclitaxel, carboplatin. And you know that this chemotherapy is quite aggressive and presents a lot of side effects like loss of appetite and other effect, inflammation for the patient. And the patients are commonly treated with glucocorticoid to alleviate these side effects. However, even if glucocorticoids are benefic for the side effect, several recent papers highlighted that on the tumor itself, glucocorticoid can have adverse effects like participating in the metastatic formation, but also in the resistance to chemotherapy and radiotherapy. So in the team, we decided to investigate how glucocorticoid and the glucocorticoid receptor have this negatory impact on tumorigenesis. And we found that that PRMT5 is really an important actor of the GR signaling. First, what we found is that in the presence of dexamethasone, that is a synthetic ligand for glucocorticoid, we have a strong increase of the interaction of GR and PRMT5. When we perform RNA sequencing to identify the role of PRMT5, the genes that are dependent on dexamethasone and on PRMT5 activity by siRNA of PRMT5, we really find that on the top genes, we found genes involved in locomotion and migration. So we perform in vitro assay, but also I will only show you the in vivo assay. We injected fluorescent TNBC cells that were treated with DEX, plus or minus invalidation of PRMT5. And we had the chance to follow the migration of these fluorescent cells in the zebrafish, you can see here in green, and measure these metastatic cells. And we demonstrated that upon DEX treatment, we have an increase of migration of the cells, and we have lost this increase of migration when we knock down PRMT5. Very surprisingly, we found that this effect on the cell migration is not dependent on the enzymatic activity of PRMT5. In fact, here we use the X-Elision technology to measure cell migration in vitro. And we found that although we have an increase of migration here, the red curve with dexamethasone, when we co-treat combined with the inhibitor of PRMT5, we don't modify at all the migration. And we confirm this result at the expression level. Based on our RNA-seq, we investigated the expression by RT-qPCR of several genes, and we found that these genes are increased upon the dexamethasone treatment, but targeting the enzymatic activity with the inhibitor of PRMT5 did not modify the expression of the gene, comforting that PRMT5 activity is not required. And in fact, what we found is that PRMT5 is more like a scaffold protein in this case that is important for the recruitment of other co-activators like HP1 gamma. We investigated also the interaction of GR and PRMT5 in a patient-derived model from Elisabetta Marangoni. And what we found is that we have much more interaction of GR and PRMT5 in the residual tumor after chemotherapy compared to the primary tumor that was not treated with chemotherapy. And when we looked also at the response to doxorubicin, we found that we have more GR PRMT5 in the resistant tumor to doxorubicin compared to the stable tumors or the regressive tumors. So this result highlighted that GRPRMT5 could be involved in chemoresistance. So we next measure in vitro in cells gamma H2AX, that is a marker of DNA breaks. And what we found is that when we treat the cells with doxorubicin, we have an increase of the breaks, DNA breaks. And when we treat at the same time doxorubicin plus dexamethasone, we have a decrease of the number of breaks. So showing that I told you before that GR is involved in the resistance to chemotherapy. And of course, we lose this effect when we knock down GR, because GR is the receptor of the dexamethasone. But interestingly, we also lose this effect when we knock down PRMT5, or here, when we inhibit the enzymatic activity of PRMT5. So based on this result, we decided to investigate what was doing the doxorubicin on GR signaling. So what we find, a very interesting result, using a polar screen assay, that in fact you have a fluorescent dye and you are able to measure the decrease of this fluorescent if your molecule will bind the ligand binding domain of GR. So here we have positive control dexamethasone and pregnenolone that are two ligands for GR. And we tested several chemotherapy. And you can see that here in green, doxorubicin is the only chemo that is able to bind directly GR at the ligand binding domain. So we carry on this study, and we found that, like dexamethasone, doxorubicin is also able to trigger GR nuclear translocation. Here you can see it by immunofluorescence, but also by subcellular fractionation. So in fact, upon doxorubicin treatment, we really trigger this nuclear translocation at a lower level than dexamethasone, but still we see a nice nuclear translocation. So based on this nuclear translocation, we wanted to investigate whether doxorubicin can mediate a kind of transcriptional program. But unfortunately, when we investigated the transcriptional activity of GR with a luciferase assay or by RNA sequencing, we couldn't really find any gene that were deregulated, or the transcription level activity is very low for GR compared with dexamethasone. So we don't think that the effect of doxorubicin does not impact GR transcription. Next, we found that doxorubicin, so I told you it triggers the interaction of GR with PRMT5, but also we find that upon doxorubicin treatment, we find that we can observe methylation of GR by PRMT5, because you see we can decrease this methylation, knocking down GR, knocking down PRMT5 also, but when we knock down PRMT9, that is an under-member of the type 2 enzyme, we don't see this decrease of methylation. So GR is methylated by PRMT5 in the presence of doxorubicin. And this methylation is specific to doxorubicin, because when we studied this methylation upon other chemotherapy, we don't see any increase of GR methylation. Interestingly, when we investigated this methylation of GR, we can also measure this methylation in the nucleus of breast tumors, in some of them. And we are now investigating the expression of this methylated GR in several cohorts of breast cancer, and the results are, the statistical correlation are in progress. So in fact, in conclusion, so for the PRMT1 part, we still carry on the subject trying to impede the interaction of alpha SARC. But if we focus more now on the PRMT5, as I told you, it's very interesting, because a lot of people only focus on the oncogenic action of PRMT5, but you have to have in mind that PRMT5 can have also good effect in the luminal tumor, especially because they mediate the tamoxifen anti-tumoral activity, but maybe also for other anti-estrogen, and it can be an important marker, a predictive marker. So in the team, we are currently now investigating the expression of PRMT5 in other cohort of breast tumors to really validate this effect, because we only have validated on 2 tumors. It's not enough. We really need to carry on this investigation. The idea will be to propose it at a new prognosis marker at diagnosis to better orientate the treatment. And for triple negative breast cancer, I showed you that glucocorticoid are able to increase the interaction of GR with other co-regulators with PRMT5, and this is involved in the side effect as a cell migration, and this effect is independent of PRMT5 catalytic activity. And now for the resistance to doxorubicin, I told you that we know that we have a complex that binds DNA, but it doesn't seem to regulate transcription. I don't show you result today because it's an ongoing work, but we have a lot of clues showing that this complex could be maybe involved in DNA repair. And we have identified key new partners of PRMT5 and GR that is involved in the repair and could explain why doxorubicin triggers this complex in the nucleus to try to modify the DNA repair pathway. So I have, of course, to thank all the fundings that help us to perform this work, and also my fantastic team. Also some collaboration with Johan Jouet for the statistical analysis, Stéphane Giraud for the screening that is ongoing, Julien Ablin for the in vivo experiment in the zebrafish, and the long-standing collaboration with Dr. Marangoni from Curie Institute for the PDX model. And I thank you for your attention, and I'm ready to take questions.
Okay. Let me see if I... Sorry, if anyone has any questions for these presentations, I think it's very good. It's a lot of experiments, and it's quite comprehensive. The two mechanisms and multiple pathways. Yeah. No, actually, I myself has questions. It looks like so you said this not figured out how actually the tamoxifens and then the PRMT5 work together. As what's the molecular mechanisms of like causing the shattering of the PRMT5 from the cytoplasm into the nucleus? Do you have any like speculations? Maybe another proteins involved? Yeah.
Yeah, of course, it's our main subject for the moment. It's of course nothing is really known in the literature about the regulation of PRMT5 shuttling. So we are using large-scale proteomic approaches to identify new PRMT5 partners that could be involved in this shuttling. Because I told you it doesn't contain any NLS. MEP50, that is its main partner, does not contain any NLS either. So first, we have to investigate how PRMT5 translocate in the nucleus without any NLS. So that's why we perform the large-scale analysis. And also to understand when it stays in the nucleus, what makes it staying in the nucleus. Because, you know, I told you that as it possesses 3 NES, each time it goes in the nucleus, we think it goes out more quickly than it goes in. And in fact, we really need to understand that's why we want to perform proteomic approaches, because we think that the proteome of PRMT5, it can be very important to really explain how this protein is translocated or not in the nucleus. So it's still under investigation. We have a candidate, but for the moment, I don't want to speak about it because it's still under investigation. We have already one candidate that could be involved in this, but we want to search for new candidate because I don't think there is only one protein. It can be a very complex mechanism.
Thank you. Yeah. Yeah, if we don't have any question, maybe we should end here. Hang on a minute.
Okay.
Yeah. Thank you very much, Dr. Rumensis, for such insightful presentation today. And thank you all the audience for members for joining us for this webinars. There are more exciting Medical Express webinars coming up soon. Our next confirmed speaker is Dr. Meisan Naimi from Ohio State University, who will talk about the CRISPR's AAV-based CAR T cells engineering for cell therapies. And so please stay in tuned for that. So at Medical Express, we are committed for fostering scientific communications and accelerating the pace of scientific discovery. We thank you again for joining us today. So stay connected, and I look forward to seeing you all very soon. So goodbye for now. Thank you.
Thank you.